Good afternoon, everyone, and welcome to week 11 of live interaction sessions for basics of crop breeding and plant biotechnology. I am Prashasti Pandey, PhD and PMRF Fellow from Department of Botany, Banaras Center University. And if you have attended previous sessions, in these live sessions, we aim to solve the questions of previous year's assignments and hope that they will help you in solving this year's assignments. And we will just solve some questions. There will be some questions on the screen. I'll give you some time to solve them. And then we can go through the concept of that question and try to understand them. Okay? So I hope this is clear. Right. So let's go on to our first question. Which of the following is incorrect for totipotency? The options are ability of a cell to differentiate into only a limited number of cell types, ability of a single cell to differentiate into all cell types of an organism, ability of a cell to continuously replicate without undergoing any differentiation, and none of the above. If you know the answer, you can speak it out aloud or you can write it in the chat box as well. Okay, so let's understand this concept. In previous sessions, also, we have talked a lot about totipotency. So let's go through the concept of totipotency once again. Okay. So, according to this, so let's talk about totipotency first. So, according to definition, totipotency is the ability of a single cell to divide and differentiate into all cell types of an organism. Okay, so this is the main definition that any single cell, okay, should be able to divide and differentiate into all cell types of an organism. Now, if that particular cell is able to divide and differentiate into all cell types, that means that it also has the ability to form complete and a functional organism. Okay, including both. So, for example, in case of plants, okay, so embryonic tissues. Like roots, shoot, or leaves in the plants, if they are getting formed, they are forming complete individual new plant which is complete and has all the function and is also functional. Okay. There is another example in case of animals like extra embryonic tissues like placenta. Okay, so placenta also has the ability of totipotency, but in case of plants, almost all the parts of plants are totipotent. Now, there is a fundamental concept in plant biology, especially that every living plant cell is totipotent. This is exactly what I was telling. That in case of animals, not all cells, very a few cells, have this ability of totipotency that it can make an entire organism. Once that organism is made, the cells lose their ability of totipotency, and now in some animals, they still have the potency of regeneration, like in case of lizards, but they cannot produce the entire organism. But for the case of plants, a single plant cell can produce an entire living and fully functional new plant. Okay, so if this is possible by some the presence of right conditions, like there has to be some nutrients and some hormones in presence of which a single cell can de-differentiate to form a callus and then re-differentiate to generate a whole plant. Okay, so this ability is called as totipotency. Okay. Now, this is why is it important to talk about totipotency? Because this is the basics, and it is a basic requirement of plant tissue culture for micropropagation and even for genetic transformation experiments, like certain overexpression lines, or if you're generating a new plant which has introduced some genes and you want to make multiple copies of it. Okay, so in that case, you can in this case the entire cell can produce a new plant, and that is why it is important. Okay, so now, as I was talking about earlier, that in case of plants, it is very evident and very uh, prominent process. But in case of animals, the protopotency is very limited. And it is usually found only in zygotes or some early embryonic cells up to 8 cells. Embryonic cells are basically up to 8 cell stage of structure. And only in these cells, there is the ability of totipotency where an entire new functional organism can be generated. Other than that, the cells become pluripotent. Okay, so pluripotent means that they can form many cells or tissues, but not all tissues. So entire new organism cannot be formed, but there are certain. So, for example, our skin. Okay, so if the skin gets damaged, it is repaired. So new skin is produced in that place. So that is the tendency of pluripotent. That it can form multiple other tissues, but not the entire organism. So they become multipotent, or sometimes they are unipotent, that they are able to form only one particular organ, but that tendency is not called as totipotency. Okay. Okay. So when we were talking about the leaf example, there is a small example that you can understand it with. that a leaf cell, when it is placed on a culture medium with I told you that there has to be some nutrients and some hormones in presence of which this pluripotency can work. So the hormones are usually auxin and cytokinin. So in presence of these hormones, a simple leaf cell should can first form callus, and from that callus shoots and roots are developed, and then it will become into a complete plant. Okay, this is the process of plant tissue culture. This is the basic uh, flow chart of plant tissue culture. Okay, that first a single cell is taken and then formed a callus. From that callus, rooting and shooting takes place, and then it gets converted into a complete plant. Now, the key features of totipotent cell is that, of course, that it can generate an entire organism. It will have complete genetic information, and it will undergo de-differentiation and redifferentiation to form an entire plant. Okay, these are the three key features of totipotent cells. So now coming back to our question, which of the following was incorrect for totipotency? The first was ability of a cell to differentiate only into only limited number of cells. The first statement itself is wrong. That it totipotent cells do not differentiate into a limited number of cells. It can differentiate into any number of cells. Second is ability of a single cell to differentiate to all cell types. Yes, this statement is absolutely correct. C was ability of a cell to continuously replicate without undergoing any differentiation. Yes, that is also possible. In case of callus formation, the cells do not differentiate into other parts, but rather they first keep on continuously replicating without going any differentiation. So C is also correct, and the process of no use. So the option A is correct in this case because it was talking about incorrect option for totipotency. So option A is correct. Okay. I hope this concept of totipotency was clear. We have already talked about this in previous sessions. Okay, so let's move on to my next question. Globular structure is observed in which phase of embryo formation? Options are inductive phase, post maturation phase, embryo development phase, and maturation phase. Okay. So let's discuss about the embryo formation first. Okay. So the embryo formation, or also called as embryogenesis. So embryogenesis generally proceeds in four steps. Okay. So the first step is induction phase. In induction phase, the cells basically acquire the embryogenic competence. Okay, this is the first phase where the cells will acquire this embryogenic competence. That is, they know now that they have to undergo this process of embryogenesis. Okay. Then second is that once those cells know that they have to undergo embryogenic process, they start the embryo development phase. So once the embryo development phase starts, there will be formations of certain structures until it gets formed into a complete embryo, and then basic structure of a new plant. Okay, so while the process of early development phase, there are certain stages which are like globular stage, or like heart shaped stage, or torpedo stage. Okay, so or torpedo stage. So these are the stages that will the any embryo will undergo while developing into a complete new plant. Once the embryo is completely formed, then comes the maturation phase. So in this maturation phase, the process of storage of the food for it to become a complete plant will begin. Then desiccation tolerance will begin. So these will all come under the maturation phase, and then it will come a post maturation phase where the embryo or the seed that is being formed will germinate and develop into a whole new plant. Okay, so this is called as post maturation stage. Now if you look at this figure, so the embryogenesis can be in two steps, in two types. Okay, first is a zygotic embryogenesis. So zygotic embryogenesis will be when a zygote is formed. That means there is a sexual reproduction. In this case, a male and a female gamete is involved to form a zygote, and that zygote will undergo embryogenesis. So how this will happen, that once a zygote is formed, after zygote, one cell structure is formed which will get divided into one basal cell and one apical cell, that will determine the direction of that plant. Okay, like apical cell will be on the above side. It will in future get developed into the shoot part, and the basal cell will get developed into the root part. That is how the direction of that cell is determined. Okay, then it will proceed to forming a 2 to 4 cell structure where the upper part, the proper embryo part will be formed, and in lower case, a small suspensor will be formed. Then these 2 to 4 cells will get divided into 8 cells, also called as octant, where the cell numbers will double. Okay. After this octant stage is the structure which is called as globular structure. Okay, where the number of cells have divided now, and it will form a prominent small sphere structure above the suspensor, and that is the reason why it is called as globular structure. Then comes the heart structure. Okay. In case of heart structure, the shoot apical meristem, also called as SAM, so it will start protruding towards the inward side. Okay, from both the sides. So if when it starts inverging from this side, the structure looks like a heart. Okay, so that is why this stage of embryogenesis is called as a heart shape, or like heart structure of the embryogenesis. And from here, the this particular point, which is shoot apical meristem, will start forming into a shoot, and the lower part of this particular cell will start forming into root, which is basically called as root apical meristem. It will start developing into root. After heart stage, there comes the structure of torpedo. Okay, so in torpedo, this invagination that has formed previously in heart shape will become more prominent. The two cells from the side will start coming upwards, and this structure will later get formed into cotyledons. Okay, and the root stems are here on the like basal side, from where the roots will start forming, and then it will get developed into a bent cotyledonal structure where this part will develop into a shoot, this part will develop into a root, and these structures will basically form the cotyledon of the plant. Okay, So this is called as bent cotyledonary structure. So this is zygotic embryogenesis in case of plants. The other structure is the other way of embryogenesis is somatic embryogenesis. So, in case of somatic embryogenesis, this is a form of asexual reproduction. So, there is no requirement of male or female gametes. Rather, any so in previous question when we were talking about totipotency, that means that any plant cell will be able to form the complete new plant, right? So, in case of somatic embryogenesis, the ability of the plant to do so is basically utilized. Okay, so there can be so explants are basically the plant tissues which are used for somatic embryogenesis. Now, there are certain important qualities that tissue of that plant should have for it to be designated as explant. Foremost, the important one is that it should be free of any kind of virus. So, virus or bacteria or basically any kind of infection. The reason is that if you take an infected cell and develop more and more plants of it, the resulting plants from that explant will also have same disease. Okay, so that is why the explant that is used, it is made sure that it is free of any kind of disease, any kind of disorder, because the offsprings from that explant will exactly be the same of the explant that is being used, and the cell should be regenerative. So mostly a shoot tip or a single plant cell or a leaf tissue or an immature embryo are used as explants. Now, these explants are taken directly into somatic embryogenesis. One step is it can directly be taken for somatic embryogenesis, or the other one is that these explants can be used to generate an embryogenic callus. Okay, embryogenic callus will then proceed for somatic embryogenesis. So it is an indirect way. Um, the benefit of generating embryogenic callus is that once this callus is formed, it can be cut down into multiple small pieces and multiple plants can be generated from each part of the callus. Because, as I was talking earlier, that callus structure is just differentiated, just replicated cells of the explants. Okay, so they have not developed the ability to specialize in anything. So all these cells have similar ability to develop into plants. So if we develop a callus from explants, all the pieces of that callus, all the parts of that callus have the same ability to get developed into a plant. So that is why this is indirect method of getting into somatic embryogenesis, where multiple plants can be generated, or these explants can be directly taken to somatic embryogenesis. And the process then goes same, that a globular structure is formed, heart structure is formed, torpedo is formed, which is finally gets converted into a cotyledonary plant. Okay, so these are the two ways of embryogenesis. One was zygotic embryogenesis, and the other was somatic embryogenesis. Now, our question, that globular structure is observed in which phase of embryo formation? So yes, it is observed in embryo development phase. Okay, not in inductive phase, because that's when the cell gets to know that now it has to go for embryogenesis. Post maturation is like after the plant has already developed and now it is going to get germinated. Maturation phase is when all the cells are getting matured in that stage. So the embryo formation phase where globular structure is observed is option C, which is embryo development phase. Okay, so I hope this was clear. Moving on to our next question: which of the following plant growth regulators promote somatic embryogenesis? The options are 2,4-D, kinetin, gibberellin, and BAP. Okay, so this is very easy question. So it is talking about plant growth regulators to promote somatic embryogenesis. Okay, so somatic embryogenesis. So there are four um, terms that was there in the options. Okay, so let's understand the function of all the four so that it will become easy to solve. Okay, so the first one is 2,4-D, which is also called the full form of 2,4-D is 2,4 dichlorophenoxyacetic acid. Okay, and it is a type of oxen. So this is important part that 2,4-D is a type of oxen, right? So what are the main functions of oxen? That oxen can induce the differentiation of plant cells. It can promote callus formation and then trigger somatic embryogenesis, especially at higher concentrations. Okay? The mechanism is that because 2,4-D basically mimics as a natural oxen, that means its functions are also similar to natural oxen. So, therefore, it can activate genes which are involved in cell division and embryogenic competence. Okay, this is how 2,4-D actually works. It will create a stress like condition and that will push the cells to go into embryogenic pathway. The most important use of 2,4-D is in tissue culture for initiating the embryogenic callus formation. Okay, this is the function of 2,4-D. Second was kinetin. So kinetin is an example, is a chemical form of cytokinin. Okay. So now again, the function will depend on the function similar to cytokinin. So what does cytokinin do? It will promote cell division, which is also called as cytokinesis. Then it will induce shoot formation, which is the part of organogenesis. Organogenesis means formation of different organs. Okay. So cytokinin will first promote cell division and then it will induce the shoot formation. Also, it has role in delay of senescence or delay in aging. Right now, what is the mechanism? That it will work antagonistically with auxin. This is an important part that it will work antagonistically with auxin. So, if there is a high concentration of cytokinin, that means there will be a low concentration of auxin, and this will lead to shoot development. Okay, when there is a high concentration of auxin and low concentration of cytokinin, in that case, shoot development will not happen. That will lead to callus formation. So, in case of tissue culture, first all the cells are the media that is given for callus formation will contain high amount of auxin and low amount of cytokinin. That will lead to generation of callus. And once the callus is formed, the callus is then transferred to a media containing high amount of cytokinin, which will now start the process of shoot development rather than proceeding the cell and continuously taking it for callus formation. Okay, so that is what makes its key use for root for shoot generation in tissue culture. Okay, so option C was gibberellin. Okay, gibberellin is also called as gibberellic acid. So the main function of gibberellin is to promote the stem elongation, to break the seed dormancy, and stimulate germination. Okay, this is done by production of alpha amylase. And then also it can enhance flowering and fruit growth. So basically, gibberellin is required in the later stage of plant when the plant has it has developed prominent roots and prominent shoot, and now it needs elongation of the stem. Or in case of germination, where the seed dormancy has to be broken, or because it helps in breaking of seed dormancy, it will also stimulate the germination. And in later stages, when flowering or fruit growth is required, then also gibberellin plays its main role. The mechanism is again by activating genes that are involved in cell elongation and enzyme production. Um, Gibberellin or gibberellic acid is not usually used in tissue cultures. The reason is that it will inhibit somatic embryogenesis. Okay, so this is why. Because it will inhibit somatic embryogenesis because it will promote differentiation. Okay, uh, it rather than helping in the formation of embryo or promoting the embryogenesis, it will inhibit this process. Okay, so it will inhibit the differentiation in embryos or the ability of cell to become embryogenic competence. It will inhibit all those things. That is why gibberellic acid is mostly not used in tissue culture. Okay, so this was the function of gibberellin. Fourth was BAP. BAP stands for benzyl amino purine. Okay, it is again an example of cytokinin. So again, the similar functions with the previous cytokinin example, that it is a strong promoter of shoot proliferation. It can enhance axillary bud growth and it stimulates cell division. The mechanism is similar to kinetin, but it is more potent. So usually, BAP is used. Is more prominently used in tissue culture. And also, it will shift the balance towards shoot organogenesis. Okay. So again, it is commonly used cytokinin in micropropagation or tissue culture. This is a small table that I attached with the presentation, so it will become easier for you guys to remember because these are the common PGRs, like the plant growth regulators, that will help the plant to proliferate or to form callus or to form embryos. Okay, and these are their types. 2,4-D is an auxin, kinetin is a cytokinin. Gibberellin is gibberellic acid, and BAP is also an example of cytokinin and their functions and how they are utilized in tissue culture function. Okay. So now coming back to our question, that somatic embryogenesis, what is used in somatic embryogenesis? So it is mainly induced by auxins. Okay, especially strong synthetic auxins, and the example of auxin in the options was 2,4-D. It is most widely used auxin for inducing somatic embryogenesis, and it will promote dedifferentiation and embryogenic callus formation. Kinetin was cytokinin, so it will promote tooth formation. Gibberellin will help in elongation and germination, so it will inhibit embryogenesis. And BAP is also a cytokinin, so it will promote shoot proliferation and not primary embryogenesis. So the correct answer to this question is option A, 2,4-D. Okay. Now moving on to our next question. Choose the incorrect option for artificial seeds. The options are: genetic recombination does not take place, produced from vegetative seeds, contains genetic constituent from both of the parents, and contains embryo, endosperm, and seed coat. Okay. So, let us understand this question. So, to solve this question, let's first understand the concept of what is artificial seeds. Okay. So, artificial seeds are basically man-made seeds which are produced by encapsulating somatic embryos or other plant tissues in a protective coating so that they can be used like true seeds. Okay, the concept of seed is the same, that there is a small embryo. Okay, I hope it is everywhere. So yeah, artificial seeds are basically man-made seeds, and they are produced by encapsulating somatic embryos or other plant tissues. So it becomes similar to a protective coating, and then it can be used like true seeds. So in case of seeds, also the concept is that there is a small embryo or a cell which can actually get developed into an embryo once the seed is germinated, and then it is protected by a small outer covering which is called as seed coat, right? So this is the concept of true seeds. So mimicking the true seed structure is how artificial seeds are produced. Now, in case of artificial seeds, a somatic embryo or some other propagule which can develop into an entire plant is usually encapsulated in a gel. Now, this gel that is used is mostly sodium alginate. Okay, so sodium alginate can easily degrade when meets the soil or when meets the water, and then the embryo inside the sodium alginate covering comes out, gets in contact with the surface, and then it can get germinated just the way a normal seed does. Okay, and once it gets germinated, it will develop into a complete new plant. Right. So now, how just to know the basic concept of how artificial seeds are formed, they first the process is of developing a somatic cell. Okay, so that is undergoing somatic embryogenesis. So somatic embryogenesis is basically vegetative cells are used in this. That means there is no involvement of any kind of gametes in this. The vegetative cell is then de-differentiated and forms somatic embryos. Okay, then these somatic embryos are coated in either calcium alginate beads or sodium alginate beads, which acts as a gel matrix. Okay, and this encapsulation will act like an artificial seed coat. So this is, if you look at the figure, that first the shoots are basically drenched, and the shoots and the nodal segments are drenched in sodium alginate solution or calcium alginate solution, and then they are taken in a dropper and then they are dropped. So basically when they are dropped in the solution, okay, so when they are dropped in the solution they will form a small structure. Okay, this encapsulated structure which will have the shoot tip and nodal segments. So each the bead that is formed will contain a single explant, and the solution that is used here is calcium chloride. Okay, so a calcium chloride solution, the sodium alginate, um, the tissues basically drenched in sodium alginate when dropped in calcium chloride will form these bead-like structures. And each bead-like structure will contain a small encapsulated shoot tip or the nodal segment, which can later get converted into an entire plant. These beads are then washed with sterile water, and then they are stored uh, like artificial seeds for later use or generation of the germplasm exchange. Okay, it can be stored and then directly sown like seeds, and the entire new plant will be formed from this process. Now, the key features of artificial seeds are that they are genetically identical to the parent because there was the use of somatic embryogenesis. That means no gametes were used, so no recombination happened. This is why the clonal feature will be there because vegetative cells are used. It will contain a somatic embryo and an artificial coating, which is not equivalent to true seed coat. Okay, it just acts like a seed coat, but it is the not a true seed coat. Also, it will lack true endosperm because nutrients, endosperm, function of endosperm is to help the seed germinate and give its nutrition while germination. So even though there will be no true endosperm, but in certain cases, nutrients are artificially added so that it will help the somatic embryos to germinate better when they receive the media or in whichever they are germinated. So there can be two types of artificial seeds. One is desiccated seeds, which is made from embryos that can tolerate drying, and the other one is hydrated seeds, which are encapsulated in gel, and they mostly need moist conditions. Okay. Now the advantages of generating artificial seeds is that it will help in rapid mass propagation because from a single plant, a lot of somatic embryos can be generated and get converted into seeds, and the um, seeds that are germinated from that will be exact replicate of the parents. So if that is what we require, then this process is very useful. It is also useful for carrying forward elite and transgenic lines. It can propagate plants that don't produce viable seeds, and it is easy to handle, store, as well as transport. There are some limitations to this process. One is that it has low conversion rate because sometimes the embryos, even despite getting all the required conditions, they do not get converted into plants. They also require very controlled conditions for germinations, and also not all species respond well to this method. So, these are some limitations of producing artificial seeds. Now, coming back to our question, um, so the first option was genetic recombination does not take place. It is correct that because it is a somatic embryo, it is formed asexually. So no meiosis or recombination has happened. Second, it is produced from vegetative cells. This is also correct because it is derived from somatic cells, which are also called as vegetative cells. Third was it contains genetic constituent from both the parents. No, because there is no meiosis, there is no zygote formation. The genetic information is only from one parent from which that embryo has been derived. So no fertilization has occurred. So there is no clone. Only single parent origin is there. Fourth option is contains embryo, endosperm, and seed coat. This is incorrect because artificial seeds are somatic embryos. There is just artificial coating, not true seed coat, and there is no true endosperm. Okay. So the correct option is C and D because the question was talking about choose the incorrect option for artificial seeds. Okay, so option C and option D is incorrect, that it will not contain genetic information from both parents, and artificial seeds will not contain embryo, endosperm, and seed coat. It will contain somatic embryo, might contain some nutrients which can mimic endosperm, and sodium alginate is definitely not a seed coat. So option C and D are wrong. Okay. So moving on to our next question. Which type of the cells or tissues could be used as suitable starting material for haploid plant production? The options are pollen, meristem, anther, and radicle. Okay. So let's go through this question. We have also talked a lot about haploid plants in previous sessions. So haploid plants are basically plants that contain only one complete set of chromosomes. Okay, so they will only contain n number of chromosomes instead of normal two sets, which happens in case of diploids, which is 2n. Okay, so the chromosome number is half of diploid, which is n. It is derived from gametic cells like pollens or ovules, and they are genetically simpler because there is only one allele of every gene present in it, and not two as it happens in case of diploid. Now, how they are produced? There are certain cultures where anther cultures are used, in which pollens inside the anthers are used. There are microspores or exactly pollen cultures, and also sometimes ovary or ovule cultures are also used, but they are less common as compared to anther culture. Actually, it is easier to produce haploid plants from anther culture or from pollen culture. Okay. The importance of producing haploid plants is that it is useful in plant breeding. It can help produce homozygous lines easily because after chromosome being doubling, the doubled haploid plants will have same alleles and not different alleles, which can happen in case of diploid, and that will aid in genetic studies as well. So now, coming back to our question, that haploid plant production in tissue culture is how it is achieved. So, the correct answer is, as we talked, that anther and pollen are the most widely used widely used cells for this. So they are achieved mostly through androgenesis, which is use of male gametes. Okay. So option A was pollen grain because directly microspores or pollen grains can be used, and it can lead to production of haploid plants via androgenesis, and it is most precise starting material. Meristem can produce diploid clones and not haploid because they are haploid in nature. They are not gamete cells, so they are by default haploid. Anther can also be used because it contains pollen. It is an indirect method of producing haploid plants, commonly It is also commonly used for haploid production. And D was radical. Again, it is a part of embryo. So that means it will have a diploid tissue. So it cannot be used from that. So the correct option to the, because it was an MSQ question, there can be multiple answers to this. So both A and C are correct in this case. Both pollen and anther can be used as cells or tissues for starting material of haploid plant production. Okay. So the next question is, which of the following crops is used for higher alkaline production through plant tissue culture? So it's not mostly a concept based question, it's just knowledge based. So let's solve this question because it's not mostly concept based question. Um, Basically, Arabidopsis thaliana, it is used as a model plant for genetics and not for alkaloid production. Hyoscyamus niger is actually used for alkaloid production. It can produce tropane alkaloids like hyoscyamine and scopolamine. Okay. And these alkaloids are widely used in plant tissue culture for enhanced alkaloid productions. Third was lens culinaris, which is lentil. It is a food crop. Has nothing to do with alkaloid production. Fourth was Lycopersicum esculentum, that is tomato, not a major alkaloid producing plant in tissue culture context. So the correct answer to this question is option B, that is Hyoscyamus niger. Okay, it is a knowledge based question. Okay. So the next question is, which of the following is incorrect as the role of encapsulated matrix in artificial seeds? The options are maintains viability, enhanced resilience, serves as artificial endosperm, and serves as protective seed pod. We have talked about artificial seeds before, so it should be easy to answer. Okay. Since you have already talked about the concept of artificial seeds, let's just see the options for it. Option A was um, maintains viability. So it is true. Encapsulation will help in retain the moisture and the viability of the somatic embryos. So option A is correct. Option B was enhanced resilience. True, because it will provide mechanical protection to the somatic embryos instead of directly storing them, which can eventually get damaged with time. It becomes much more resilient when they are stored in encapsulated gel structure. So option B is also true. Option C was it serves as artificial endosperm. When it is not exactly endosperm, but when it is supplemented with some nutrients, then it can function. It can functionally be treated as artificial endosperm. So even if it has no similarity with the biological endosperm, it is not identical to that, but it can be treated as endosperm. And D was serves as protective seed coat. So this is wrong because although it provides protection, it has nothing to do with a true seed coat, and it is formed after fertilization. So the incorrect role of encapsulated matrix of artificial seeds is serving as a protective seed coat. So option D here is incorrect. All right. So question 8 is which of the following statements is incorrect for TI plasmids? The options are the tDNA region is flanked by left and right border. Opine catabolism region is responsible for coding the protein for the metabolism and uptake of opine. The virulence gene is located inside the tDNA, and the gene of interest to be transferred through agrobacterium mediated transformation is located inside the tDNA. We have also talked about TI plasmid in previous sessions. Okay, so let's talk about TI plasmid. TI plasmids are also called as tumor-inducing plasmids, and they are a large circular DNA molecule which is found in Agrobacterium tumefaciens, and it is widely used as a natural genetic engineering tool because this plasmid has the ability to transfer a part of gene and transfer that part of gene into the plant and integrate it in the plant's genome. Okay, so that is why it is used as a natural genetic engineering tool. Now, how does it do it naturally? So, when agrobacterium infects the wounded plant cell, during that infection, it will transfer a specific DNA segment. Okay, and that DNA segment has to be between the left border and the right border of the tDNA fragment. Okay, so whatever gene is present between the left and the right border of the tDNA, that particular segment will get transferred into the plant and gets integrated in the plant genome. In natural conditions, TI plasmid and agrobacterium, and by transferring this particular gene, causes crown gall tumors, and it will basically encounter tumors when this disease happens in the plant. It will make the plant cells produce more opines, and because of this production of more opines, that opine is now used by the bacterium as its nutrients, and it will keep on growing. So it will basically um, uh, command the plant cells to produce its nutrient, and the plant cells will produce more and more nutrients for the bacteria to thrive inside that plant, and that's how it keeps on growing. So once this bacteria enters any wounded plant, it keeps on it hacks the plant genome, and it will order the plant cells to produce that only that particular nutrient that the bacteria will require for its survival, and eventually kill the plants because it is not able to produce other nutrients that the plant actually requires. So, talking about the structure of TI plasmid, it has several important regions. The most important one is tDNA, or also called as transfer DNA. So, the any gene that is present in this tDNA will get transferred into the plant cell, and it is flanked by a left border and a right border sequence. And in natural TI plasmid, this tDNA will contain tumor formation genes and opine synthesis gene, because this will get transferred into a plant, and once it gets integrated in the genome, that is how it gets commanded to produce opine for its nutrients. Okay, but in case of biotechnology, or in case of using it as a genetic engineering tool, this tDNA segment is basically cut. The tumor formation genes and the opine genes are basically cut, and the gene of interest is introduced. The entire other mechanism remains same. So that is how it is used for genetic engineering. The second portion is virulence region, or the Vir genes. So now Vir genes are located outside the left border, and this is the left border and right border. So Vir genes are located outside the tDNA. That means it will not get integrated inside the genome. Rather, it will help in cutting, transferring, and integration of the tDNA in the plant genome. These genes by themselves do not get integrated in the genome. They just help the bacteria for this entire process to take place. So they are called as virgin. There are multiple virgins, and each virgin has different function for this entire process to take place. Third is opine catabolism gene. Again, this is this gene is not present inside the tDNA region. So if you notice here closely that there is one opine written inside the tDNA and the other one is written outside the tDNA. Now the difference is the one that is integrated inside will, which is there inside the tDNA, will get integrated in the plant genome and help the plant in producing that opine. But the other one, that is opine catabolism gene, this will help the bacteria to utilize that opine that the plant is producing as its nutrient source. Okay, so the third part, that is opine catabolism genes, will help these bacteria to utilize the opines which is produced by transformed plant cells. And fourth is, of course, ori, or the origin of replication. This will help the plasmid replicate inside the bacteria, which is an essential part of being a plasmid. Okay. Okay. So how it is used in genetic engineering? As I told previously, that in TI plasmid, the tumor causing gene has been modified now, and the desired gene that we want to insert, or the gene of interest, is now replaced inside the tDNA gene, and agrobacterium is used in same method to transfer these genes into plant. This method is called as agrobacterium mediated transformation. Okay. Now, why it is important? Because it acts as a natural gene delivery system and it is widely used to develop transgenic crops, stress resilient plants, and disease resistant varieties. Now, coming back to our question, the incorrect option. The first was cDNA region is flanked by left and right border. That was correct because the left and right border will determine the tDNA region and only that part will get integrated. B was opine catabolism region is responsible for coding the protein for metabolism and uptake of opine. This is also correct. Opine catabolism region will allow the bacteria to utilize the opines which are produced by the infected plant cells. C was wireless region is located outside the tDNA region, is located inside the tDNA region. So this statement is wrong because it is located outside the tDNA region and not inside. D was the gene of interest to be transferred through agrobacterium is located inside the tDNA. Yes, this is correct because only then the gene of interest will get inserted inside the plant genome. So it has to be within the tDNA borders for transfer. So the correct option to this was option C is incorrect that virulence gene is located inside the tDNA. I hope this PI plasmid concept is clear. The next question is which of the following sterilant is used to sterilize soft tissues of plant? The options are mercury chloride, calcium chloride, sodium hypochlorite, and sodium chloride. Again, it's a knowledge based question. Okay, so this small functions of all the four compounds that was given in the options. So first is mercury chloride. Mercury chloride is a strong disinfectant, but because it is its ability to act as a strong disinfectant, it is used for sterilizing hard plant part materials. Okay, like seeds or the parts which are not basically soft tissues should not be sterilized with mercury chloride. So usually while sterilizing seeds, mercury chloride is used. Second was calcium chloride. It is used to maintain membrane stability and as a nutrient source, but it is not a sterilant. C is sodium hypochlorite. It is a mild surface sterilant which is commonly used for soft plant tissues. And D was sodium chloride, which maintains the osmotic balance. So again, it is not for sterilization. Uh, it is rather used for balancing the osmotic, basically maintaining the osmotic balance. Okay. So now again, coming back to the question, surface sterilization for soft tissues, we mostly use mild but effective sterilants. So mercury chloride cannot be used because it is very strong and toxic. For hard tissues and seeds, it is correct. Calcium chloride is not a sterilant. Hypochloride, sodium hypochlorite is which is also called as bleach. So it is commonly used. It is effective, gentle on soft tissues, and it is a standard sterilant used in tissue culture lab. Sodium chloride again is not a sterilant. So the correct option to this question is C, sodium hypochlorite. Okay. So the last question is, which compound is released at the site of damaged plant tissue, facilitating the entry of agrobacterium? The options are esters, phenolics, ethers, and ketones. Okay. So let's talk about this question. When we're talking about the concept of how agrobacterium basically infects the plant, so this is the concept. Okay. So when the plant is basically wounded, okay, in wounded plant stem, it will release certain compounds, certain chemical compounds, which will help the bacteria to identify that the plant is now wounded and it can easily infect it. So these compounds that are released by the wounded plant is called as acetosyringe. Okay, and acetosyringe, when it binds to the bacteria, it will give signals to the bacteria to activate the weird genes, and these weird genes which is present inside the agrobacterium will now get activated, and by their activation it will strip off the single stranded tDNA from the left border and the right border, and now this tDNA will get coated in a protein coat and get transferred to the nucleus of the wounded plant. Okay. Once it gets to the nucleus of the wounded plant, it will release the protein coat. The single stranded tDNA will get integrated inside the plant's genome, and that's how the entire bacterial cycle will keep going on, because when it gets integrated inside the plant, it will help in production of opines. And because this tDNA gene has opine catabolism mechanism, it will keep getting nutrients. The plasmid will keep replicating. The agrobacterium will keep replicating, and it will form the tumor for which this particular plasmid is responsible. Okay. So the important part here was that the wounded plants will secrete some chemicals which will help the bacteria to identify this process to start. Okay. That chemical is acetosyringone. Now, acetosyringone is an example of a phenol. It's a phenolic compound. Okay. So basically, coming back to the answer of our question, that when plant tissue is wounded, it will release some signal molecules that will attract the agrobacterium and it will activate its virulence genes. So the options were first Easter, it is not involved in signaling. Phenolics, option B was correct because wounding plants release phenolic compounds like acetosbenzoate, and these will activate the Wilt genes and facilitate the bacterial infection. Again, ethers are also not involved and ketones are also not involved. Okay. So the correct answer to this question is option B, phenolics. Example of phenolics is acetosbenzoate. Okay. Okay. So I hope this mechanism of infection to plants by agrobacterium was clear. And phenolics was just a part of it. Okay. So we will end this session here and we will meet again next week, Tuesday, from 6 p.m. to 8 p.m. Okay. See you then.